early are each of you working or tapping regulators to help with trial design? Is this happening before things get started? How involved are they in the conversation?
So we are always like,
ladies first.
all right. So we currently have 2 products in the clinics. The first one is currently in phase 2B. It's being investigated for the most severe expression of vestibular pathology. It's called acute unilateral vestibulopathy. To design actually the trial, we had to discuss with the agencies very early on, and we actually created the primary endpoint. So we looked at different areas. We looked at pain because we thought it could be very relevant for our vertigo trial. And then we pressure tested with patient advocates, and we said, if we measure this, does it change the quality of life? We discussed with the doctors, is it clinically significant if we reduce vertigo by 1 day or 2 days, or etc. And then with patient advocates, we were asking them, what does it mean when you have something like that? And it was like so compelling for me to hear when some of them said, I'd rather give birth to a child without, without how do we call this, like the peridural?
Epidural.
Yeah, thank you. Rather than going for another.
I've been there, of course.
Yeah. Sure. You guys know what I'm talking about. Rather than having this acute crisis, and then you're like, okay, every day matters. Then in this case for them, they can't even stand up. So very early on, and regulatory is because even for them, they had no idea, and using parallels and patient groups and doctors.
I agree totally. When we are in that we can say specific area like immuno-oncology is new. That means after checkpoint, and checkpoint inhibitor as today standard of care, is nothing published, and our trial is after checkpoint failure. Then of course we can discuss about the endpoint of survival, but what is clinically meaningful when you have not literature in order to say that is the level? And then you discuss with expert, clinician, in order to say what is meaningful. And that is not easy to have this meaningful status. And after that, you present your meaningful vision to the agency, and they say yes, no, perhaps, but at the end, we agree about what we think in the endpoint.
I do think that it's very important to get in front of the FDA and EMA early. And sometimes when you're dealing sort of with frontier medicine, of course, it's in both parties' interest to share perspectives early. So cell and gene therapy is still a relatively new field. Mm-hmm. And, when you have 5-10 patients worth of data that already suggest you're changing the course of a disease, then I think it's also in both parties' interest to find a way to get it to patients relatively quickly, because the current trajectory of those patients is dramatic. And if you can really change that course, then time really matters. Mm. So again, I think it's a very constructive and sort of creative approach to what is required.
I think we probably all agree that it is important to engage into discussions with the regulators early on. I mean, there's no question about it. Particularly in biotech, I mean, we, for example, are in the HIV field, but we are not developing the 24th or the 25th antiviral that is actually knocking down the virus in the periphery, but is not going to the core where the disease is taking place, and this is the reservoir. If you look, there is no published guidance from either the FDA or the EMA on what should endpoints for this study, what kind of endpoints you should choose for your studies. Therefore, again, the critical interaction is the science, and actually I include clinical research into that. Today we always talk about clinical development, but as a matter of fact, we are going from data point to data point, because what we are achieving today or what we are seeing today is going to define the clinical trial that we are starting tomorrow. And that is only possible if we keep the relationships at a very high frequency with the regulators, inform them about the data we have, and then move on.
I like the clinical research thing because beyond, the primary endpoint, what we try to understand also is like how is the disease evolving, and biomarkers are also now more and more important, I think, to include, because you really know if you're impacting the natural state of the disease or not. So I think it's important.
Yeah, just to add on to what she's saying, I think it's to understand the full NDA program when you do your pre-IND meeting. And it's beyond clinical. You'll get a lot of insights with respect to the toxicology program as well. And even within clinical, there have been changes in our case with acne in terms of reducing the age of the patient from 12 years to 9 years. So it's a good learning from a point of view how perspectives are changing in US FDA, so that for us to prepare what kind of patient pool that we should be targeting.
We're fortunate in the last year and a half in hepatitis B that the two major medical societies in the field, the American Liver Diseases and the European Liver Diseases, convened with industry, FDA and EMA, a summit for 2 days in September of 2016. And a consensus panel issued a publication from that to give guidance to the industry on what will be the key determinants of functional cure in hepatitis B. In particular, the FDA, led by the division director, who was physically present with many of her senior staff and the EMA at the meeting, the 2-day conference, said, we will accept this certain marker, surrogate marker, drawn from blood, as an endpoint, acceptable endpoint for definition of efficacy and functional cure. That's very big for us, because it gives a guidance to the industry on what endpoints we need to study in our pivotal programs. So it's been very collaborative. I can't say enough about the FDA and the EMA for participating in that and giving guides. Now, you then have to go forth and have your end of phase 2, your pre-IND, your end of phase 2, and discuss your own trial design, because there's modifications given in your own therapy and what you're studying. But at least generally, we understand. So in our phase 2 program, to say, can we determine POC, proof of principle, we'll know better, even before we go into our pivotal trial designs. We're fortunate in hepatitis B.
Yes, in immuno-oncology we are facing a big revolution, because for the first time we are working on a new checkpoint domain, that means a myeloid checkpoint.
Right.
And the revolution is because we are not thinking about indication, but about type of inflammatory aspect that you can find, a cold or hot tumor. And that is completely new. And the registration, as an example, in MSE, that is the name of microsatellite instability, not limited to one cancer, but open for other cancer, is a big revolution, because that is the first time that we are not thinking about indication, but about vision of the tumor microenvironment. And that is very exciting to be in this area now.
I concur. We're fortunate in viral diseases in some way. I don't know if those on the panel familiar with the area would agree in that. Typically, when you're measuring efficacy in viral infectious diseases, they're very objective measures. You're drawing from the blood. It's hard to have a placebo effect, like you were considering pain in your trials. I ran a pain company. You can be influenced your endpoints by a placebo effect. Typically, phase 2 data is highly predictive of pivotal trial results in most viral infectious diseases, oftentimes. So we're fortunate in that sense. But now oncology is evolving.
Yes.
It's evolving right in front of us. And that keeps me up at night as we think about what will be our endpoints in our trials.
Well, but I would say the viral area is evolving as well. I mean, it's not only the measurement of the virus in the blood, because the patients that we are dealing with, they have no measurable virus in the blood. They have a reservoir where the virus has integrated its genetic material, and that is something that is sort of at a different scale measurable.
I should clarify myself, not in the case of the latent virus there, but in the case of, in HPV, there's, they're replicating massive quantities of the virus, so you can clearly measure that. Now, of course, there's a reservoir in HPV as well.
Yeah, exactly.
Is,
and that's the holy grail for you as well, I would suppose.
Right. Yep. And that was the guidance we were getting from the agency and the FA on the EMA and how to measure that. What would be the markers on measuring that, which is very helpful to us.
Exactly. And as you said, you had the discussion with FDA some time ago. We are seeing exactly the same discussion now between FDA and the key opinion leaders coming up in 2 or 3 weeks from now.
Right.
Yeah.
Right.
And I think one of the opportunities in the discussion or the dialogue with the regulators is if you've got an effect size that is significant, is avoiding the need for placebo. And, you know, finding a way to get to a registration where you're really basing that on published natural history or natural history that is well documented elsewhere, and therefore avoiding the need of putting patients on placebo. So I think that this is a really big advantage if you can find a route with the regulators where maybe you've identified some important centers which have well-documented natural history. You know, that is something that you can look at, and I think that's, you know, very advantageous in terms of speed to market.
One thing to add on this topic, I mean, I think we all agree that we would love to talk to the agency early and often, and as long as they're willing to return our calls, because there's no downside. The more we learn about their feedback, the better we can plan, the smarter we get, the smarter they get, so on and so forth. Obviously, sometimes they have other things to do. So in those cases, at least from our development timeline perspective, what we try to do, what we target is the moment we have a lead nomination, for example. So we're within a year, 15 months of an IND application. That's when we start putting together our advisory board. And that includes, of course, the physicians, key opinion leaders, but we also make sure to include former FDA personnel in that board, such that whenever we can ask the FDA for their feedback and whatever they can tell us, great. But whenever they don't know what they have in mind, you know, they would like to see more data before committing to a certain path. In those cases, we try to have individuals with similar experience who has sat on the other side of the table with us, trying to allow us to make more informed speculative decisions.